Hi, my name is Rebecca Pinals, and I work with Professor Marquita Landry at UC Berkeley. Today, I will be sharing my work in elucidating protein corona composition and dynamics on nanoparticles in biological environments. Applying nanotechnologies to probe and modify biological systems is exciting because we are employing tools that exist at the same scales as the fundamental biological processes we seek to access. For sensing, nanobiotech platforms are highly tunable, enabling quantitative detection of biochemical and physical changes at the time and length scales at which they take place. For imaging, nanoparticle-based contrast agents can enter and shed light upon bioenvironments that have been otherwise invisible to us, providing high spatiotemporal resolution images of such biosystems. And finally, for delivery, nanoparticles offer the advantages of localized delivery to targeted locations, or the ability to penetrate otherwise inaccessible bioenvironments holding insoluble or unstable cargoes. So in sum, engineered nanoparticles and nanobiotechnologies more broadly have significantly enriched our toolbox of strategies to investigate and access biosystems. However, when a nanoparticle enters a biological environment, proteins spontaneously adsorb to that nanoparticle surface, leading to the formation of what is known as the protein corona. This phenomenon is relevant for any nanobiotechnology application translated from in vitro development to in vivo use. Protein corona formation unpredictably changes the nanoparticle identity and fate as those adsorbed proteins mask original surface characteristics and endow new biochemical properties to the nanoparticle. It then becomes the nanoparticle corona complex rather than the as-designed nanoparticle that interacts with biological machinery. This in turn causes vast implications on outcomes such as cellular uptake, biodistribution, and the overall functionality of the system. Moreover, the protein corona is dynamic and displays a high degree of temporal heterogeneity as proteins rapidly bind and unbind from the nanoparticle surface. Accordingly, the protein corona is a complex system that, despite its importance, remains poorly understood. With this in mind, the central aim of my research is to develop an understanding of the protein corona and then apply this knowledge towards rational nanotechnology design. Today, I will be focusing on the first branch of my project in determining the composition and driving forces governing corona formation on nanoparticles. I will also present my results in probing protein corona exchange dynamics, but encourage you to watch my talk linked in my poster for further details. And finally, I have leveraged these fundamental findings on the protein corona formation process to design and develop new nanosensors. Most recently, I applied this corona-centric design concept towards a near-infrared nanosensor for rapid SARS-CoV-2 detection. And here I encourage you to see Talk 88E for more details. I want to begin by introducing carbon nanotubes as a nanoparticle possessing unique physical and optical properties for nanobiotech applications. Carbon nanotubes exist on the nanoscale, and their high aspect ratio provides a large surface area upon which we can load functional cargoes for delivery or sensing moieties to build a nanosensor. However, these nanotubes are composed of all carbon and are therefore extremely hydrophobic. To apply these nanotubes in aqueous bioenvironments, we employ non-covalent surface modification with biomolecules and polymers. This surface-adsorbed moiety is then tunable, with some examples including phospholipids, DNA, and protein. Single-wall carbon nanotubes are an excellent platform for nanobiotech development because they exhibit intrinsic near-infrared fluorescence as shown in this spectrum. This fluorescence emission falls within the tissue transparency window, here shown in purple, meaning that the fluorescence is transmissible through biological samples such as tissues and biofluids. And due to the semiconductor nature of these carbon nanotubes, this fluorescence is highly photostable over the course of many hours, unlike organic fluorophores that degrade in photobleach. This photostability enables continuous, real-time imaging of biological processes over the timescales required. As an example of how we implement these carbon nanotubes, we have developed a nanosensor for the neurotransmitter dopamine. This nanosensor is based on carbon nanotubes functionalized with a specific sequence of DNA, a repeat of G and T either 6 or 15 times. Nanosensors are generally designed and optimized in vitro to yield a significant fluorescence modulation upon analyte binding. As such, the presence of dopamine elicits a large increase in the carbon nanotube fluorescence, shown here imaging nanosensors on a glass slide exposed to dopamine. Yet, when we apply these nanosensors in vivo, spontaneous protein corona formation often leads to a diminished or abolished nanosensor response. This is true for nanotechnologies applied across all potential bioapplications. In this case, our dopamine nanosensors retain their response to evoke dopamine release in mouse brain slices. However, this response magnitude is drastically reduced. More broadly, protein corona formation presents a critical challenge in effectively translating our nanotechnologies from the test tube into these actual biological systems. Today, I will share my research involving the characterization of the protein corona formed on these carbon nanotube-based nanosensors, with a focus on the corona composition, driving forces, and dynamics. First, I will present results on the corona composition formed on DNA functionalized carbon nanotubes in blood plasma. And importantly, this framework is generalizable to varying nanoparticles across different bioenvironments, though today I will just focus on my carbon nanotube results in plasma in the interest of time. And I probe the composition here by employing a selective adsorption assay and then characterizing those bound proteins by mass spectrometry-based proteomics. I have done quantitative liquid chromatography tandem mass spectrometry to build a map of the protein corona composition formed on DNA carbon nanotubes in blood plasma. And here I will build the dataset piecewise. First, I'm showing proteins on the y-axis and relative fold change on the x-axis. Spot size denotes molar abundance of each protein. Returning to the x-axis, fold change is relative to plasma proteins as they exist in the native biofluid. This line at 0 would then indicate the same amount of protein adsorbed in the corona phase as exists in the bulk biofluid phase. Therefore, greater than 0 indicates enrichment in the corona phase, and less than 0 indicates depletion. I can next classify proteins into functional roles by color, and we begin to see this distribution of enriched, highly abundant proteins across different functional roles, such as complement proteins and lipid binding proteins. Finally, I want to highlight some top binding proteins of interest, specifically apolipoprotein A1, complement C3, and fibrinogen. Interestingly, although albumin is the most abundant blood protein, it is so depleted within the corona that it does not fit on the left-hand side of this chart. So to summarize this map, we find that protein corona formation is selective, not merely a function of native abundance of proteins present in biofluids. And we begin to see some key corona proteins emerge, which we can then choose as case studies for downstream testing and which I will circle back to later in my talk. With this knowledge of what proteins adsorb, I next wanted to understand the driving forces governing this adsorption. To gain a more in-depth understanding of the interactions driving corona formation, I began varying the incubation parameters of the blood plasma with nanoparticles and characterizing those bound proteins by two-dimensional gel electrophoresis. First, we have all of the proteins present in plasma alone, with the darker color shade indicating a higher frequency of appearance. The reference state incubation is now with nanoparticles, done in a static condition at ambient temperature with biologically relevant ionic strength. Next, a dynamic incubation is used to probe corona stability. With constant dynamic motion, only more tightly bound proteins stay associated with the nanoparticle, thus providing a proxy of adsorption strength and insight into the hard, tightly bound versus soft, more loosely bound corona proteins. These dark purple proteins are now posited to represent the hard corona. Varying ionic strength is used to probe charge effects. Elimination of salt increases the contribution of electrostatic interactions due to the absence of charge screening. And we begin to see this trend emerge where proteins present under dynamic conditions, so the hard corona proteins, are also present in the absence of electrostatic screening. Thus, electrostatic screening seems to facilitate formation of the soft outer corona. And finally, higher temperature incubation increases weighting of the entropic contribution to the overall free energy change of binding. Proteins still present under higher temperature either display an entropic gain upon adsorption or an enthalpic driving force. So to summarize, these hard corona proteins are essentially able to bind under all of these variable incubation conditions, despite this being electrostatically unfavorable. And therefore, we posit that hydrophobic interactions drive these hard corona proteins to adsorb. And finally, just to reiterate these key corona proteins that we already began to see with the mass spec data, ApoA1, complement C3, and fibrinogen are key hard corona proteins. I next wanted to explore what the time-dependent corona behavior looked like and gain a more holistic understanding of the exchange occurring in bioenvironments. Towards this end, I developed an experimental platform to measure exchange of biomolecules on the carbon nanotube surface. We take advantage of a known photophysical property of carbon nanotubes where fluorophores in close proximity to the surface become quenched. We label our entities of interest, here protein and DNA, with distinct fluorophores, and then we can measure binding exchange by multiplex fluorescence monitoring of both molecular species on the carbon nanotube surface. The assay workflow is simple and high throughput, where we inject these two solutions together in a well plate format and measure the fluorescence with a fluorescence plate reader. And as an example of the actual output, we can use fibrinogen as a model high binding protein. Upon injection of fibrinogen, DNA originally on the carbon nanotube surface desorbs, as shown by this increase in fluorescence as the DNA is removed from the carbon nanotube surface. Simultaneously, fibrinogen adsorption occurs rapidly, where a horizontal line at 1 would indicate no adsorption. Returning to our plasma protein corona map, we can then choose a panel of proteins to test with this assay. And based on these end-state corona values, we might expect protein adsorption if the proteins are enhanced and abundant in the end-state corona, so on the right side with a large spot size, as compared to low binding expected if the proteins are depleted and in low abundance in the end-state corona, so the left side of this map with a small spot size. I next apply our corona exchange assay for each of these individual proteins. Here, I am monitoring Cy5 tag DNA desorption from the carbon nanotube surface as a function of time, using this as a proxy for protein adsorption. I can summarize these single protein experiments and compare to the end-state protein fold change from mass spec as shown here. The x-axis represents the protein fold change from the compositional studies, and the y-axis summarizes the DNA desorbed from the carbon nanotube due to protein adsorption after 2 hours from the corona exchange experiment. We see two populations emerge in agreement with our previous hard and soft corona groupings, one with significantly more DNA desorption and thus more protein binding than the other. Interestingly, certain proteins deviate in both rate and end-state binding. Complement C3 is extremely abundant in the end-state corona, yet displays less DNA displacement than we might expect. We can summarize that in general, the end-state corona formed in the whole biofluid is indicative of individual protein binding rates and end states, yet deviations from this trend suggest cooperativity effects involved in protein corona formation, as demonstrated by complement C3. Just briefly, we have shown that this corona exchange assay is generalizable to measure dynamics of other biomolecules on the carbon nanotube surface. Circling back to the dopamine nanosensor I introduced at the beginning, I want to show how this corona exchange platform was used to develop better nanosensors. After passivating exposed nanosensor surface with phospholipids that are linked to the hydrophilic polymer polyethylene glycol, or PEG, we demonstrated a sizable reduction in protein adsorption, as I'm showing here with this bar graph for varying molecular weights of the PEG moiety. This anti-biofouling property then resulted in a more robust nanosensor response in mouse brain slices, as well as reduced morphological changes imparted to mouse microglia. To summarize, by probing the selective protein adsorption to nanoparticles, I was able to quantitatively map protein corona composition, identify key corona proteins, and then investigate interactions driving the corona binding. Beyond the end-state corona, I also developed a corona exchange assay and applied this assay to measure in-solution real-time corona binding dynamics. This demonstrated cooperativity effects that are present in full biofluid scenarios, absent in such single protein experiments. And finally, I briefly alluded to how we can implement the corona exchange assay and this corona-centric framework more broadly in terms of nanosensor design. And I've done this by harnessing these innate protein carbon nanotube interactions to create a rapid CoV2 nanosensor. Now I want to end my talk by reiterating that formation of the nanoparticle corona complex challenges our control over the projected nanobiotech application. Yet corona formation concurrently presents an opportunity as a functional handle to tune and enhance different nanoparticle properties. This corona-centric framework will be impactful in biological sensing, imaging, and delivery applications, where taking this corona-centric design approach from the beginning will allow us to design better nanobiotechnologies. With that, I would like to thank my entire lab, the Landry Lab at UC Berkeley, especially my advisor, Professor Marquita Landry, who has been absolutely fantastic, as well as the whole team of people involved on these various projects. I'd also like to thank our funding sources, and specifically the Women in Chemical Engineering Committee for the Travel Award. And thank you for listening, and I'd be happy to answer any questions.